My topic is wet weather, purpling, canola diseases, and their impact on the yield. I have outlined my topic as a little bit introduction, wet weather, purpling, research done at LREC, results, and summary. I'll show you the situation we had over there here in especially in northeastern North Dakota last year. It was very wet. We in May we received around 6.36 inches of rain. In June, 5.27. In July, 2.91. So, and August, 4.35. So on. It's a lot of rain, and it's what wet weather does. This is how some of my trials looked. It's very wet. They are all drowned, and on top of it, I had we received more rain. You see, my canola plants have emerged over there. On top of it, more rain, more rain. I lost all these trials over there, and because of that weather, we have found we got severe root rot infections. It also farmers also reported from different counties, so we went and checked. Uh, lot of them. You see this over here, and wire-like structures on the top right hand. Uh, this is what we saw due to wet weather. Then severe white mold infections, you can't imagine. Sometimes I felt like whether they were sprayed or not. If they were sprayed and we saw this much of infections, probably there is another thing we have to take care of. Fungicide resistance? I don't know. But yeah, that's what we saw. And again, because of severe rains, we saw some of the clubroot resistant varieties of canola gave up, and they are no longer resistant. You see this picture on the left side. It is at flowering. We saw severe wilting and death. And when we pull the plants and the roots, we saw galls, severe galls, and each gall can produce billions of spores. Also, severe lodging. Lodging can be caused by several things. Here, we thought late season lodging is because of heavy rains and winds, and severe pod shatter due to late season rains and winds. You see this, but I don't know whether they are pod shatter varieties or not, but this is what we saw over here. And then purpling and canola. Often this is also we saw, and a lot of some of our growers over here were reporting that we have been seeing purpling in canola for the last 3-4 years. It's a regular feature over here. uh, then I did a little bit of literature search. In that we found that it's a clear indicator of nutrient deficiency. That's what we all thought, but in fact it's a complex response to various stress. I feel like whatever it stresses, whenever a canola plant is under stress, it shows this purpling. In the affected areas, the plant do not produce chlorophyll that allows the underlying purple pigment to become visible. So this is what happens. So what are the factors which can make a canola plant to get under stress and express this purpling? Nutrient deficiencies of sulfur, nitrogen, magnesium, boron, phosphorus. So all these can act. If they are individually, each nutrient requirement is specific for the canola plant. If it is less, it could show that kind. Sometimes in a drought stress, 2023, because of drought, we might have seen, or can be a herbicide, or it's a herbicide carryover damage, or pathogen infections, mostly root rots or insects. So everything, all lot of factors that can create purpling in canola. Some of the examples I would show you. This is a 2023 picture which I took in west of Cavalier County. It was dry, and you see the purpling. That's because of drought stress. And this is a picture which I got from Australian website. It says under heavy insect pressure, you can see purpling. It's a severe purpling over here. And this is a Canadian Council publication where they showed that after 60 days they can some if they are deficient in nitrogen, canola plants can show purpling. Likewise in phosphorus, the phosphorus deficiency in canola. So yeah, every nutrient, every stress condition can create purpling. Look at this picture. In the left, you have the cupping and light mottling, and then in the lower leaves are purpling. This is because of sulfur deficiency. Even sulfur deficiency can be this couple like leaves can be purple later. So similar kind of symptoms are exhibited when it is stressed with boron, boron deficiency. So the yellow mottling, cupping, and purpling common for both nutrient deficiencies. So in our areas, whenever we see purpling, when we used to think that that is because of sulfur. So nowadays, yeah, we have to figure out what is what. These are the couple of examples of herbicide. This is the picture on the left is herbicide carryover, and the picture on the right is glyphosate damage. So that's what it's both are from Canola Council. This is how it looks. Everything shows purple, even if it is carryover or herbicide damage. Purpling due to disease infections. This is look at this nice picture on the left side. It says yeah, it's a complete purpling. It is caused by turnip yellow virus. Luckily, it's not here, but we haven't seen this kind of symptom. This is from Australia. This picture, so we don't have that disease yet. This is on the right. It is the blackleg canker. On the This is a canker caused by blackleg fungal pathogen. On top, you see the purpling of the stem. So this is common in here too. This is purpling due to disease. And more purpling we saw this year due to disease in root rot complex pathogens. What are the root rot complex pathogens? Are Rhizoctonia, Fusarium, and Pythium. So this exclusive Rhizoctonia can cause purpling on the stem, and this light purple, light it's not reddish, little bit reddish or little bit uh, brownish yellow. This is caused by Fusarium. So we took this and this root rot complex can be caused by Pythium infections early in the season. It makes entry to the other pathogens like Fusarium and Rhizoctonia into the system of the plant. And this is the same pictures all the way in the right side down are received from Rolette County, where Anita has collected these. And you see this one of the plant over it is a wire-like. In pathological terms, we call it a wire-like symptoms. This is what it causes. Root rot complex causes. Then the root the root becomes wire like. At flowering it exhibits and it can detach from the main stem on above the ground. So then you will see the death of the plants at flowering. So all were seen for the first time in my 11 years of experience because of wet weather. When we got those plants to the lab and we isolated different pathogens. This is Fusarium, this is Rhizoctonia, and this is Pythium, where all three matches the root rot complex, which could, and you see this picture. This is a pretty nice symptom of girdling and the wire-like symptoms when we pull out the stem out. How do we manage root rot complex? Very challenging. Partial resistance available. Cultural practices like diverse crop rotations, practice very longer rotations. Fungicide treatments can improve canola emergence and yield. In all, integrated approach is needed. Then the next disease we have seen is what psyllium stripe. It's a potential threat to our canola industry. It is there for several years, but we identified in 2020, and then since then we are working on it. It is caused by a fungal pathogen, Verticillium longisporum, infects brassica crops mostly, That's the good thing. Yield losses associated with verticillium stripe are between 10 to 50%. In 2021, we saw severe lodging. A lot of farmers, I attended a lot of farmer calls of lodging, and we gathered the symptoms, the stubbles, and brought it to the lab and identified that it is caused because of verticillium longisporum. What are the basic symptoms here? Half yellow leaf yellow, half leaf yellowing. Not always, but literature says half stem browning. But this is the perfect example of verticillium stripe in a standing crop. Half of the stem will be green, the rest of the half will be brownish yellow. And when it's at the swathing or a little bit further, when you go and pull the stem out and you see the peeling of the epidermis or the outer layer, underneath you will see black pinpoint strike structures called microsclerotia. That is the perfect symptom of verticillium. And added to verticillium, and blackleg also can infect the same plants. When both the verticillium and blackleg pathogens interact and infect canola plants, the losses might be severe. Verticillium stripe? How do we manage verticillium stripe? Longer crop rotations, no resistance varieties available, no fungicides available. So we are trying to find some management options over here. First, we started with annual survey. We have 3 years of data now. Seed treatment evaluations, we are trying 2 years of data. I did varietal screening. We did some varietal screening, and there is a statement. The PhD student in Canada, Alberta, he did a research on high pH favors verticillium stripe. So for that, we have some products which we can compare whether really the pH is influencing uh, high pH is influencing what psyllium stripe incidences. This is where most of my surveys we did. After swathing, we go to the field, we collect the stubbles walk in a W pattern, collect the stubbles and cut at the crown region and see whether it has verticillium infections or not. At the same time, we collected soil sample. Remember in the last slide, I mentioned that pH is influencing or not. So we sent these soil samples to Fargo soil testing lab. They has given us the data. My 3-year survey observations are here. In 2022, 21% of the fields we visited has verticillium with an incidence percentage of 18. You remember 2022 is a average year in precipitation terms. 2023, 70% of the fields has verticillium, but the percent of incidence is only 2. It is a dry year. 2024, 94% of the fields we visited has verticillium with an incidence of 29%. So prevalence means presence of the disease in a field. Percent incidence means number of infected plants out of the total stems we have evaluated. So it is there in our, in a wet year like last year, it can flare up. So that's what we have seen. But if next year it's a dry year, we might see a very less incidence as we saw in 2023. Remember, I was mentioning what we did with the soil samples. We got the pH data out of it, and we correlated with the incidence data, which we got over 18 counties. And this is what I got. It is negative response. That I can say that we have to do more correlation studies to see what really happens. Is it really high pH is influenced? But for now, in my data, it says not really. High pH is not influencing verticillium incidence. The verticillium stripe varietal trials we plant at the research center in a replicated trial, and we have around 12 varieties which are commonly used by farmers. This is how we rate at the end of the year. Here is my data. out of the varieties which are commonly used in our area by the farmers. InVigor L255 is an old one, and I have InVigor L233P. It's also a old one. It's not in, but the rest are all in the market. With this, I can show that in verticillium index is on right side, vertical axis, and likewise I put in on the left side yields. So the grayish bars are yields of each variety. You see this InVigor L350 PC. I got a 2,369 pounds per acre, and the lowest is in 255 PC 1400. So there is a difference among the varieties where to choose. There is a varietal difference. But again, it's a 1 year data. I have to test for a couple of years more to figure out apples to apples. And then verticillium stripe seed treatment trials. So since verticillium longisporum, the fungal pathogen, is a soil borne, we have to approach with the seed treatment managements. So we have tried these. There is an experimental seed treatment which did very well. Saltro, which is very common, which we treat farmers use for blackleg in Canada. Then the common one, Evergold Energy, Intego Solo, Rankano Summit, Trilex, and compared with the non-treated. We have here also we have good differences. You see again verticillium index on the right side, horizontal axis, vertical axis, and yields on the left side, vertical axis. Uh, Experimental, I got around 2,525 pounds per acre. Then none of the varieties, none of the seed treatments matched it. Unfortunately, this is not registered, so we won't be seeing it. We have to only pick among the rest of the seed treatments. Again, I have a couple of years of data, and each year it's different. Like last year, it was dry. But still, I have good infections. This year, we got nice up to 50% of verticillium index in non-treated. So, but there are options we are having that we can go for. Rancona Summit did very good over here. So, if 2-3 years of data, I can put forward what are the seed treatment chemicals that can work on verticillium stripe. The next big disease in our area is clubroot on canola. It's a soil-borne disease caused by lower organisms. Everybody knows for the past 12 years we are doing this research, and it's when it infects in a large scale, 100% yield losses is possible. Here again, what we do at LREC is survey varietal resistance and soil amendments. Visual surveys where we saw the drying and death of the plants, lodging, and these are the galls we have seen. In our clubroot survey, you see then 2000 we have done 12 years of research over the surveys. In 2018, we got very peak period. In 2019, resistance varieties were introduced into the system. So on, we got lesser infections. But this year, the resistant varieties gave up, and we got around 10 out of 23 fields scouted had clubroot. What are the varieties? This is the first report of breakdown of resistance in resistant varieties. We have four different cultivar characteristics by the different industries. First generation varieties, we have saw 100% of losses. So these CR4 varieties, we have saw 100% DSI, but it's in patches. And the third one is resistant to currently predominant pathotypes. It also has occurred in patches. The next generation varieties show fewer galls. I don't think the resistance has occurred over in this, but in the rest of the three varieties, I have seen resistance breakdown. This is how it looked in field conditions. How to manage clubroot? go for 1 in 2 instead of going for 1 in 2 to 1 in 3 year rotation. Can drops the spores by loads by 90%. Current successful rotation here in especially in northeast and North Dakota is canola, wheat, either field pea, soybean, sunflower, or dry bean after wheat. Then go for wheat, then canola. It will work, and use of resistant cultivars highly recommended. um, Blackleg, it's generally a number one disease in states, still the one, and we have tested several um, seed treatment chemicals. Again, the experimental did good. Then the next best one, Indigo Solo and Saltro. White mold, it's another, since it's a wet year, we have seen a lot of white mold. I have shown the pictures. Luckily, we have a very good forecasting system in the state. If you visit, which is if you visit the site created by NDSU, NDON, and NCGA, that will create a risk calculator, and you can go for a fungicide spray at 20 to 30% of flowering, which could be very effective. So this is my trial where we did, and Sprolin is still working. I got an 1850 pounds per acre last year. The next one are there is a difference between the chemicals too here, and here is my non-treated sec. I got only 965 pounds per acre. With this, I would like to summarize all the disease incidences had a significant effect on yield last year because it's very wet. Accurate diagnosis is essential to manage these diseases. For blackleg, rotation of varieties and longer crop rotation is working well in North Dakota. White mold, a single spray of fungicide at 20 to 30% of flowering is very crucial. Clubroot first generation resistance varieties are no longer effective. Currently, longer rotations with rotation of multi gene resistant varieties will be helpful. Pathotype study is currently ongoing. Verticillium stripe is prevalent issue in surveyed counties of North Dakota. One canola in 4 years, that is a very long rotations to be practiced. Seed treatment and varietal resistance seem working, however, needs extensive research. My final advice to the grower is grow multiple canola varieties in a year, can spread out the risk and provide some insurance to the canola. Thank you. I'll be happy to answer questions.